Good evening, everybody. Thank you so much for joining us. And Isabel, Christina, Rob, thank you so much for bringing us together. It's my great pleasure and honor tonight to introduce Dr. David Johnson and Hui-Chun Su Johnson, whose pioneering research on soil microbial communities points us along a pathway for simultaneously addressing climate change, food security, water security, and several other of the most pressing challenges confronting our species. Their talks and presentations around the world are transforming agriculture around the world, and tonight it's our pleasure to have them here right with us. Without further ado, Dr. David Johnson and Hui-Chun Su Johnson.
Thank you, Patrick.
Hi, everyone.
This will be the Cliff's Notes version, okay? 20 minutes is not a lot of time. So I will go ahead and get it started.
We reside on this beautiful living planet. I invite you to let me take you on this journey through understanding how the biological processes and countless iterations of evolution have influenced the path to Earth's current state. With this understanding, we can begin to transform our agroecosystems from degenerative to regenerative and improve our soils, our environment, our health, and the livelihood of rural communities. Let's begin. Microbes dominated almost 80% of the time that this planet has been developing. It is important to understand that microbes are the backbone of every organism and ecosystem on this planet. No organism can survive without its resident microbiome. First, we have to look at how nature has set this up. There's a ladder of succession on plant systems that goes on between bare soil parent material up through your old-growth forest. As you move up this ladder, you see a change in productivity from about 90 grams of dry biomass per square meter up to 2,200 grams of dry biomass per square meter per year. What accompanies that increase in production is a change from a bacterial dominant system to a fungal dominant system, and also from a low carbon content to a high soil carbon content. Right now, we're about here in our agroecosystems, bacterial dominant, low carbon, low production. We grow weeds very well. The weeds have a purpose on this planet. They are the organisms that begin to transition poor, broken soils up this ladder of succession and are good indicators of soil health. We need to be in this range, about 1-to-1 on your fungal to bacterial ratio. That's where a succession of grasses, row crops, and vegetables do well. We also need to understand that agriculture is controlled disturbance. In order to stay on that rung of the ladder, there's a certain amount of disturbance that has to occur. Nature developed a unique system about 53 million years ago. That's when ungulates or grazers evolved along with grasslands. They both appeared in the fossil record about the same time. Nature's system on the Great Plains involved bison. The bison were kept in tight herds by predators. They had to move around regularly so they could only graze about 40% of the forage that was there. The other 60% was trampled into the ground. They would deposit their dung, their urine, and their saliva. The dung would be rolled up in small balls by dung beetles and placed underground. Basically, it would be composted in place. Perfect temperature, moisture content, soil microbiota, and worm communities, allowing a maturation process. What was going on as the bison moved was a microbiological inoculation event that swept across the prairies. So how can we mimic this system on agricultural lands? Not everybody wants or can have bison or cattle on their property. My wife, Hui-Chun, and I came up with a no-turn composting process that mimics nature. This composting process results in a fungal-dominant, biologically diverse compost. The bioreactor mimics what happens to dung and organic matter in the soils of the prairies. The conditions for composting are always aerobic, approximately 70% moisture content, no turning, and you never let it freeze. This bioreactor is up on a pallet. We have 6 air columns coming up through the center. The pipes are pulled out after 24 hours as the pile completely stabilizes itself with newly developed fungal hyphae and actinomycetes. Worms are added after the thermophilic phase when the temperature goes back down below 80 degrees Fahrenheit, and it takes about 1 year to reach maturity. We started out with greenhouse trials, comparing our compost, the tube on the left, to 8 of the compost available in our area. We observed the amount of plant growth, the blue bars on the graph, that the compost promoted. As you can see, our compost promoted twice the growth of any of the other composts. Nitrogen, phosphorus, and potassium are the key components that are supposed to indicate the potential for plant growth. It wasn't so in this test. Our compost was very low in nitrogen, phosphorus, and potassium compared to the other compost, and yet we had over double the growth of any other compost. We thought, okay, it's organic matter. That's the key. Yet again, not so. Organic matter is important, but it did not correlate with plant growth. We had measured the fungal to bacterial ratio along with system physical chemistry, and we noticed the fungal to bacterial ratio was a very strong predictor of plant growth. These findings promoted a follow-up greenhouse trial with green chili, exploring how the fungal to bacterial ratio affected plant partitioning of carbon and nitrogen. We looked at what percent of photosynthate was partitioned in the root carbon, the brown piece of the pie, the carbon that went into the plant shoot, green part, and the carbon that went into the fruit, the red part. New soil carbon, over and above what soil carbon we started with, in gray, the carbon that it was respired, in blue, and the flow of carbon into nitrogen fixation, the orange part. As we go from a bacterial dominant low carbon system here on the upper left to a fungal dominant high carbon system over here on the bottom right, we see a change in the percent of carbon that's flowing to the plant. In a bacterial dominant low carbon system, only 3% of the energy the plant captured through photosynthesis actually went into the plant. The rest of it went into increasing soil carbon, into respiration, and towards nitrogen fixation. As we move up the ladder to higher fungal to bacterial ratio, the amount of plant photosynthate increases to 17%, 21%, 30%, up to 56% of the energy captured in photosynthesis going towards the plant. We are here now in our agricultural systems. We're about 11% efficient. This may indicate that we should be able to increase our productivity on these lands about 5 times. Another thing we observed in this greenhouse trial was the relative soil carbon respiration rate, or a measure of system carbon use efficiency. Notice in the bacterial dominant systems, low carbon, up to 55% of the available carbon in that system was respired. When you move towards more fungal systems with higher soil carbon mass, only about 11% of the available soil carbon was respired. Complementing the higher biomass production observed in fungal dominant systems was a significant increase in system carbon use efficiency. After our greenhouse experiments, small plot trials were initiated. Inoculation rates for these trials started at 400 pounds per acre in a one-time application. As you can see on this white square, it's just a dusting of compost. So the compost we used as a microbiological inoculant, not a nutrient resource. This trial explored the potential of plant growth on desert soils, one that had no legacy issues from previous agricultural practices. Cover crops were grown on this soil, harvested, hauled off, and then another cover was planted. This was to explore the capacity of an inoculated soil microbial community's potential to promote plant growth. On the left, you can see that the beam, or the biologically enhanced agricultural management treatment, produced almost twice the biomass as the control in the first winter cover crop. If you look in the background, you can see how good the soil was. It is a desert area, so the soils are very poor. This is a second winter cover, using the same seed mix, where over 1,600 grams of dry biomass per square meter grew. This is the third year's winter cover crop. 2,190 grams of dry biomass production and over 588 pounds of nitrogen fixed per acre, all with only a one-time application of compost, growing covers one after another and hauling off all biomass production. Winter cover crop productivity increased from 492 grams to 2,244 grams of dry biomass per square meter in this 3.5 year experiment, demonstrating a 4.6 times increase in productivity, very similar to what was observed in the greenhouse experiments. This winter cover crop is planted in October or November. It idles through the winter about 2 inches high and then wakes up about mid-February, and it grows to the end of April. In 10 weeks, the plant growth equals the productivity of the most productive ecosystem on the planet when it has the right soil microbiology. The summer and winter cover crops together produced about 3,186 grams. Remember, the most productive ecosystem on the planet is 2,200 grams, so half again more than the old-growth forest. This is what the soil profile looked like when the experiment was started. All sand, no A horizon. There was a 3-inch A horizon developed in the control, so even cover crops help. When the microbial inoculant is implemented, it seems to kickstart the system, and we observed a 7-inch A horizon develop in the compost-treated soil over that 3.5 years. Field trials were started in 2010. On the left, you are looking at our control. On the right is a biologically enhanced agricultural management treatment. This was a 4.5-year project looking at how fast we can increase soil carbon. Compost is applied only once, at the beginning at a 400 pound per acre rate. From there, covers were grown year after year, mowed down, lightly disked in, and then the system was replanted, so all biomass grown was left on the plot. After a year of beam, a winter cover crop produced 250 grams of dry biomass per square meter, compared to only 50 grams of dry biomass in the control. You can see this is a 5 times increase. 7 years later, on the same trial plot, you now see 1,144 grams of dry biomass per square meter grown, which is almost 5 times the amount observed after the first year. This chart shows how the soil carbon increased over the 4.5-year duration of the project. Soil carbon increased from 0.4% to 1.5%, for an annual increase of 0.24% in the top 30 centimeters of the soil. This rate is equal to 10.7 metric tons of carbon per hectare per year, or 37 metric tons of CO2 per hectare per year. This is a comparison of beam with other long-term carbon research projects. Most other researchers see only 0.2 to 0.7 tons carbon per hectare per year potential to place carbon into the soils of agroecosystems. Promoting functional soil microbiomes promotes over 10 times increase and the potential for a 19 times increase in advanced beam. Comparing the production of conventional agriculture with beam systems, over twice the growth was observed in both cotton and chili. It is the microbiology that needs to be promoted in our farming systems to bring them up to what we're seeing in a biologically enhanced agricultural management approach. Farm-scale corn production field trials were next. 3 separate treatments were implemented on a half circle in Wilcox, Arizona. A conventional approach with 256 pounds of nitrogen per acre, a beam plus 15% of the nitrogen, or 38 pounds of nitrogen per acre, plus 2 pounds of the Johnson Su compost applied as an extract injected into the furrow at planting. And a third treatment, beam only, using the 2 pounds per acre of compost as an extract injected into the furrow at planting. This is what the results looked like the first year. The beam plus 15% nitrogen matched the productivity of the conventional nitrogen approach and was $121 an acre more profitable. The beam only had 2% yield loss and was still $86 more profitable than the conventional approach. The following year, we implemented the same treatments growing pinto beans. In these trials, the beam plus 15% nitrogen outperformed the conventional approach by an extra 5 bushels per acre, or about $85 per acre more profit. The beam only outproduced the conventional approach as well by almost 3 bushels per acre, for $47 per acre more profit. It appears once you adopt beam and start cutting back on fertilizers, the soils seem to become healthier and more productive each year. What was noticed in a parallel experiment was the difference between injecting the extract into the furrow at planting and spraying the extract on top of the soil after planting. The difference was significant. Over double the plant growth was observed when the extract was injected into the furrow as opposed to a topical treatment. A follow-up experiment was conducted on a summer cover crop harvested in the fall. The sorghum plant on the left had extract injected into the furrow, and the one on the right had a topical treatment. The in-furrow treatment reached reproductive maturity, where the topical treated failed. The difference between treatments was a successful crop and a crop failure. These are the results from a farmer in Belgium using a seed treatment of Johnson Su compost on a multi-species cover. He saw the exact same results on his cover crop, a doubling in productivity. As a reminder, our use of compost as a microbial inoculant in cropping systems, we are trying to mimic what's happening in natural grazing systems. Let's briefly look at how this is implemented in adaptive multi-paddock grazing management systems. These are results from Gabe Brown's ranch. He has increased his soil organic carbon from 1% to 6.5% in 20 years. For the first 13 years, from 1993 to 2005, no-till, crop diversity, and cover crops enabled Gabe to achieve an approximate 0.1% increase in his soil organic carbon. When he stopped using nitrogen fertilizers and reached about 3% soil organic matter, started using multi-species covers and eventually livestock integration, this rate of soil organic carbon began to increase to approximately 0.56% soil organic carbon increase per year. This is what his pastures looked like when we visited. For the previous 3 years, he had had 5 inches of rain per year. It's not a lot of rain, but his pastures had forage that grew over the hood of his 4x4 truck. Gabe has relayed that he has seen a 30 times increase in the productivity of his pastures. In a project with Peter Bick at Arizona State University, we are comparing adaptive multi-paddock managed ranches to continuously grazed ranches in the southeastern U.S. in a full systems analysis. Our portion of the research was to compare the influence of these two approaches on soil biology. The adaptive multi-paddock, or AMP systems, demonstrated an increased photosynthetic capacity of 93.1 grams of dry biomass per square meter. The AMP systems also had almost 0.5% higher soil organic carbon. The AMP systems also demonstrated a 35% reduction in the relative rate of respiration, or increased system carbon use efficiency when compared to continuously grazed management. AMP systems also provided higher carrying capacity, allowing 2.24 times more animal units per hectare. So practicing the grazing management that mimics the bison on the Great Plains helps to regenerate the microbial community population, structure, diversity, and biological functionality of these grazing systems, increasing photosynthetic capacity on the front end, reducing the carbon flow out of the system on the back end, and these two attributes together help increase soil carbon. There's now a push to implement carbon capture in the soils of agroecosystems. The carbon we put back into the soil needs to be a biologically functional carbon, carbon in the form of microbes that fix nitrogen from the atmosphere and furnish it to plants, and also microbes that break down the soil parent material to gather essential elemental nutrients and shuttle them to plants in a form of biological barter in exchange for photosynthates that contain sugars and proteins. Our current greenhouse gas emissions from burning fossil fuels is about 36.8 billion tons of CO2 per year. That is about 3% of the total amount of CO2 flowing into the atmosphere. CO2 emissions from our current agricultural approach accounts for approximately 17% of those fossil fuel emissions. The other 95% of CO2 that flows into the atmosphere results from respiration from microbes in the soil, our ocean, and respiratory processes from plant metabolism. We have a fantastic opportunity to draw down carbon dioxide from the atmosphere by not only reducing agriculture's fossil fuel footprint, but also by utilizing the carbon capture potential residing in the interplay between soil microbiomes and plants to restore carbon back into our soils. Globally, there are approximately 1.25 billion hectares of temperate grasslands similar to the AMP ranches we studied in the southeast U.S., and another 1.36 billion hectares of farmland. Our current soil systems are broken, and we will not be able to accomplish carbon offsets of any magnitude in the soils of our agroecosystems with a business-as-usual approach. What is necessary is to adopt practices that offer improvements in efficiency for increasing photosynthetic capacity, to increase the amount of carbon flowing into the system, and those that improve carbon use efficiency on the back end to slow the flow of carbon out of the system. What amount of carbon offsets could our previous observations offer if adopted on these lands? The data from our field research in the ranching sector indicates an extra 18.2 billion tons of carbon dioxide could be pulled from the atmosphere per year through increases in photosynthetic capacity and carbon use efficiency in the soils of the 1.25 billion hectares of rangeland on this planet. Our current research results in the farming sector indicate up to a 5 times increase in photosynthetic efficiency with a doubling of crop productivity over current conventional practices, and an estimated 35% improvement in soil carbon use efficiency. Estimating carbon capture potential on only 50% of these rates would yield another 27.5 billion tons of CO2 pulled from the atmosphere per year on the 1.36 billion hectares of arable land on this planet. For those of you who say we can't do this here, this is Alejandro Carrillo's ranch in the Chihuahuan Desert in northern Mexico. When he took over management of this ranch about a decade ago, it took him 250 acres to support one cow. He normally gets from 6 to 7 inches of rain on a good year. He started practicing adaptive multi-paddock grazing on his rangeland. This used to be grasslands hundreds of years ago. You can see the 3-foot soil carbon profile, but overgrazing destroyed these systems and desertification took over. And now he's completely changed that environment from desert back to grassland. And now it only takes 20 acres to support one cow, so with proper grazing, he's increased his photosynthetic capacity 10 times. Another example, Ian and Di Haggerty are farmers in Western Australia. They farm about 20,000 hectares of dryland barley, and they have stopped using fertilizer and are applying the microbiology using compost as an extract injected into the furrow at planting, at the same rate of application that we do. On their barley crop, they get about 3 tons of barley per hectare in a dryland farming area that conventionally gets about 1.5 to 1.6. You can see the rhizosphere developed on the roots of the barley seedlings. What is most interesting to me in this picture on the left, they have already harvested the barley in this field, but with late rain after the harvest, you see a C4 grass sprouting. This grass had not been seen in over 4 decades, sprouting up in nice, neat rows exactly where they injected the compost extract when they planted the barley. Application of these microbes seems to reawaken the soil seed bank. This is another interesting thing we came across on Facebook. It is also from Australia. A rancher sprayed some of the Johnson Su compost extract on his rangeland. He could only spray on one side of this tree as the branches were too low on the other. It's quite astounding to see that one side of the tree leafed out like that, and it leafed out almost a month before the other side of the tree did. This is the difference in the chestnuts between the treated on the left and the untreated on the right. So we can meet the challenges we face. We can rebuild degraded soils and increase soil fertility. We can produce more nutrient-dense food, use less water to grow crops by increasing both water storage capacity in our soils and plant water use efficiency. We can reduce our energy use by allowing nature to extract or fix essential nutrients. Reduce agrochemical pollution and atmospheric CO2 at the same time, but most importantly, we can help ranchers and farmers improve productivity and profitability. I would like to thank the Howard G. Buffett Foundation, the Thornburg Foundation, the Globetrotter Foundation, and the No Regrets Initiative, the Mighty Arrow Family Foundation, and the Laird Norton Family Foundation for making this research possible. Here are some links you can get more information at the Center for Regenerative Agriculture and Resilient Systems at California State University, Chico. It has all the information on how to build the bioreactor and use the extract, and the observations of compost makers and users worldwide. Thank you very much for the opportunity to share this research.
Okay, we're back.
Good. Great. Okay, so now we move to the question part of the evening. And comments are welcome as well. Let's see, Pat, I think we wanted you to lead off with the first question, please.
Sure. So, David and Hui-Chun.
Go easy on me, Patrick. I know where you live.
True enough.
We want to say thank you to David in particular because this is our Earth Day event edition of Soil Stories. Go ahead, Pat.
Yeah. Any guidance on doing this at the home scale?
Don't hesitate. Do it. Don't talk about it. Just go ahead and do it. Get in the mix on this. We've had so many people say, gosh, that's a lot of work. But the rewards are overpowering. when you see the results.
And I want to add this, is that usually most of you gardeners or people who have green thumb, or you have access to a lot of organic matter in the fall. And you can get all those yard trash and leaves and things like that and get it built. And you don't even have to build a full-size one. You can cut it down to maybe 2/3 or half of the height. And then you can get plenty for your backyard.
Yeah. Patrick
And
has done this.
yeah, and you build it, you just look after it by observing, make sure it's water, make sure it's not frozen. And for the rest of the time, you don't have to go out there and turn it. You don't have to do anything to it. It's almost like, you have a garden and then you put on a sprinkler system, put on the timer, and then a year later, that you have this wonderful product.
Now start the learning process as soon as you can. We need your input. We need everybody's observations on this.
David, I understand you have a registry of folks who are doing this?
Yeah, Chico. Chico has a registry of all the people that are participating. And, we get comments from them. We get ideas from them. They get questions answered. It's just nice as a community. Yeah, it's kind of working like that quorum sensing where all the organisms work together collectively for the benefit of the whole. And, that's what we're striving for here, is to have this take off. We see it in about 60 different countries right now.
Wow. Okay, great. Have you done any research on this, using the compost on urban trees, David?
Well, I've done it on my trees at the house. I have some cottonwood trees that they were getting a disease, and I just pulled the bark back to the cambium and spread the compost on it, and it took care of it. And that's just an anecdotal evidence, but we've seen it on pecan trees. We've also seen it on almond trees, that it benefits the productivity. And jujubes, also quince. We saw one lady in California that put it on her quince trees, and she had quince the size of grapefruits when her neighbors were the size of plums. So there's something about how this works. We don't know how it works completely.
So if it works on the other trees, I would imagine for urban trees, it should work. Although I do feel sorry for a lot of urban trees because they don't get a lot of soil around them for percolation of the water. And a lot of time they kind of bound by this concrete area. So yeah, that's a totally different subject, I suppose.
Okay, great. And Allison, you had a question about Alejandro's process. Would you like to unmute and ask their question?
Sure. Thank you for that really interesting presentation. And yes, I was just struck by Alejandro Carrillo's really dramatic results, and just sort of wondering, could you walk us through and give us a little bit more detail on how what he did and how long it took to produce those results?
Well, a lot of this is herd impact. Having the cattle the right amount of time on that soil, not too long, enough time and enough density to get a cow patty about every square meter, and to have the hoof action to break that crust up and to push the organic matter in the soil. His recommendation is to start in the best areas and work your way out, because he says that's the best place to really see the gains. And that also allows you to start bringing that seed bank on the hooves of the cattle into the other areas as you're improving those areas. So it didn't happen overnight. This happened, I think, over a decade for Alejandro. But he's completely restored that desert area. And he's seen some of the growth on those plants up to his shoulder when he's sitting on a horse. So there's a lot of impact that these animals have that we just have really completely disregarded. If you want to see a lot of good videos on this, carboncowboys.org Peter Bick's site. He has about 10 videos there that show all the different people that are using this adaptive multi-paddock grazing management and the benefits that they see.
Yeah, and also go back to Alejandro, is that since this is in Chihuahua Desert in Mexico, he was able to use, just have a couple of cowboys to herd the cattle. And this is actively managed. So it's not just leaving the cow out there on their own and, you know, kind of meander around, eat whatever they want. They actively manage their cattle and herd them from places to places, and actually have the observation of the land as far as what the amount of forage, what kind of forage there is. And so how long they will leave those cattle on that piece of land and move on to somewhere else. And they assess the amount of rainfall they have so they can understand or know how quickly those forage will recover as well. So there's a lot of observation and active management involved with that.
There's also a certain amount of selection of the breeding of the herd. He wants to have cattle out there that will also eat the brush, and have the mothers out there that will teach the young cows also the same traits. So he's very observant on the herd and what cows that he culls out of it in order to pull that system forward.
Okay, now Allison, we have a number of people in New Mexico who are managing similarly. So if you want to get in touch with us, we could potentially direct you toward them. David Flogger, you have your hand raised. Would you like to ask your question? Unmute, please.
Yeah, hi, can you hear me?
Yes.
Yes.
I'm just wondering, I live in the north in Michigan. I'm wondering, you said you can't allow the bioreactor to undergo a freeze. How could you do that in the north and not have it freeze?
Well, we've had some people put it into dairy barns with the dairy cattle. The dairy cattle kept it warm. If you had a shop that didn't freeze, that's another alternative. A root cellar is ideal if you know of one or have one. You could possibly hay bale it and maybe put a heat tape under it just to keep it, you just need to keep it above freezing so the worms won't die on you. We've had one person in Alberta actually build an anaerobic composting pile that they had put tubes through to circulate water, and they control the heat of the anaerobic pile with a fan and pushing air into it. And that kept their greenhouse and fish pond and everything thawed through 45 below. So it's just how innovative you can get on this.
Okay.
Remember, this is a biological incubator. So these are living organisms. So freezing is not conducive for them to thrive.
It can go down to 32. We've seen that in a experiment in central Colorado where they used a hoop house and built a 30-foot by 40-foot bioreactor in it. And it got down to 32, but when we opened it up, the worms were still crawling around. So, and it looked really good under the microscope.
Cool.
Great.
Thank you.
Sure. Let us know if you figure out a different way.
Share with
Okay.
everybody.
Greg Corning, you want to unmute and ask your question?
Sorry, I got so caught up, I sort of forgot my question. It's in the chat somewhere. You can
Yes.
go to the next.
You ask if the bioreactor can be built without petroleum-based landscape fabric, like someone else suggested, maybe burlap. Will that work?
I'm not sure how long the burlap would last. You could try it. I know in Africa, they're weaving straw or reeds to build that container.
Or bamboo, because I mean, we didn't really appreciate the terminology dirt poor until when people told us about it. And just, it warms your heart to think about those people, how innovative and creative they are. They decided we can weave, and so we are going to make this this container that is going to hold all this material for 1 year, and also able to allow the air to go through, allow this bioreactor to breathe, but then yet not too much airflow to dry out the outer area of this compost bioreactor. So yeah, there are some pretty innovative people that when they really want to do it, they come up with some creative solution. So may it be keeping it from freezing or keeping it from drying out and still holding there with a different container design. But as far as the woven landscaping cloth that we came up with, is also through trial and error. Because remember, this incubator needs to stay intact for at least 1 year for its maturity. So if you use material that is going to degrade on you prematurely, that you have a problem for the maturity date. So just keep that in mind.
And you need a certain porosity as well.
Yeah.
If it's any consolation, Greg, that we have microbes in there that degrade plastic. So it's amazing what these organisms can break down.
Okay, thank you, David. Let's see, John Kreitzman, you had a question?
Yes, thank you for the presentation. I'm a retired civil engineer and mine reclamationist, and it seems to me that this approach has promise in revegetating drastically disturbed lands, such as road cuts and mined lands. And I'm wondering if there's any research that you're aware of or trials going on in that kind of scenario using this approach.
I know a lot of the mine reclamation processes do use microbiological amendments. This one is particularly beneficial from the other ones we've seen. To see that seed bank in Australia, 40 years they hadn't seen that C4 grass. And for it to pop up, I mean, the people that worked with the government there didn't think it was native to the area, but the old timers that had herded sheep knew that it was native to the area. So, again, there's a lot we don't know and a lot of impact that this can have. And it would be nice to see it trialed on some of the mine reclamation. I mean, if anything can do it, we know it takes the biology to do it. And it seems like what we're putting in there is a roster of microbes. And when we do metagenomic analysis, nature doesn't choose all of them. She'll choose a select few, but they're different in different situations because it's a dynamic system. So again, there's a lot we don't know and a lot of experimentation that needs to be done.
Thank you for your question, John. Jeff Anderson, he was asking David about water needs for things in the desert to grow, and commented that or asks whether the pictures that you showed were taken during the rainy season, and what might it have looked like during the dry season.
Oh, okay, well, in the experiments I have done, I do not use water as a variable. I want to see what the capability of this is. So these were irrigated plots. But the thing to remember here is when we use the water, we want it to be efficient use. We want to get more crop per drop. And this is what this system does, is if you have the microbiome there to work and function for the plant growth, then you get more biomass, more growth out of the system. You can actually double crop productivity because that would enable you to use only half the land to grow the same amount of crop on the same amount of water. So, you have to look at this from a different perspective. How can we make this system more efficient? Because we waste a lot of water trying to grow crops in a poor soil, and the dynamics of cutting water use in agriculture by half, since they use 70% of the water on the planet, the fresh water, is will be quite impactful.
And I just want to add this. Neither the beam biologically enhanced agriculture management nor the adaptive multi-paddock grazing are magic practice, but they are practice that mimic nature. And trying to go through this way of work with nature is instead of working against nature. And I believe the question that the gentleman asked about those two pictures, the one might be during the dry season, the other one is the raining season. Based on what I understand on that project, where we got the picture from, that for the research project, that it was a process of that one picture of a dry land that was 10 years ago, the other picture was 10 years later. And it's through the process of this actively managed adaptive multi-paddock grazing managed system that bring it back to that grassland looking thing. But you know, it's not overnight, it's not magic.
I did one more thing on the water use. Well, I was going to add, I know, I've forgotten what it was. I'll come back with it.
Okay. Let's see. In the interest of time, because I'm looking at about 21 potential questions from you all, and I would like to get through as many of them as possible, I'm going to go ahead and ask some of these questions to move things along.
Okay.
All right. So Natalie is asking about research in orchard systems, and I believe you kind of addressed that in comments on fruits earlier. Let's see.
Except
And you've talked about.
I don't know what technique specifically would be used. Would it be the actual compost through foliar sprays, or this injection thing, which I don't even know how that works? I don't even know
Okay.
what that means.
I'm glad you asked that. You need the biology, but you also need plants. So you need to have a cover crop system to feed the microbes. They need the energy that the plant is capturing. So if you can have cover crops within that orchard, and the best way we see is to make an extract and inject it into the furrow at planting. That seems to be the most effective. We've seen people coat the seed, and it did pretty well in Belgium. We were very surprised at the results in Australia on that one tree, that chestnut, on the chestnut tree.
Yeah.
And you know, to see a whole tree over there without a trunk, what kind of strange in that picture.
So the takeaway from that picture for me is that you need to have living roots, living organism enabled for those microbes to collaborate and collaborate with. Just like us human, you can have all the talent in the world, and somebody take you and drop you off in a desert and leave you there by yourself. None of your talent is not going to come to fruition, because you would die and parch in that desert. So you need something to work with, somebody to work with.
Josh Bowman in Las Cruces here has done it in a pecan orchard, and he's combining both the inoculation with cover crops and grazing sheep, and that's turning out very effective. He's cut back on the amount of water he uses. He actually grows more pounds of pecans on each tree. They're larger nuts, they have better fill, so he gets a better price on them. There's a lot of benefits as you start to bring the biology back into the system.
Great. Natalie, thank you for clarifying that. It was a really important point. Linda Poole asks if you have any experience in using this approach to recover biodiversity of native plants. She's in northern Montana, and they're looking at an issue with crested wheatgrass.
Well, just looking at what Australia did, if you go ahead and read the rest of what the guy did, what he noticed is the cattle would only graze where he had sprayed the compost extract. And he had gone out into the field. He normally goes out into his field to gather cow patties for his tomato in his garden, and the cow patties were all degraded. So there's a lot of other things going on here.
Bring back the living, healthy biological system is very important.
Chris, why don't you ask your question? I'm not sure I fully follow what you're asking there. Chris Sheehan?
Yeah, sure, no problem. What do we do about existing, okay, if we're not planting something, but we have existing perennial cover crops, existing grasses that we might like to build up the quality of our soil, what technique would we use? Because I know, I've watched a whole bunch of your presentations. In general, spraying is not as effective as in furrow, but if you're not actually planting something, you have existing plants that you want to improve the mycorrhizae and whatnot, what techniques would you suggest for improving more open areas that have cover crop, but maybe you're not necessarily adding new seeds at that time?
Yeah, well, we haven't done that research, and it's on the agenda, but to see what happened in Australia, I was very surprised to see that kind of response, spraying it on the soil. I'm not exactly sure how he did that. But from the research that we've done, we see that spraying on the ground wasn't that effective on annuals. It may be more effective on perennials if you have a living root in the system.
Already.
Yeah,
it's already there. It's ready to do something.
Yeah, that's the thing. And timing on that would probably be a key issue, either before a rain or in a morning dew, something like, so you can get some extract soil contact.
Great.
Chris, we'll let you do that experiment. I'd love to see what goes.
Sounds good.
Okay. Good or bad,
Okay.
I still need to know.
I have a clarification. The question is, if it gets down to freezing accidentally, does it totally kill it, or does it just pause it and lengthen the finish time so that freezing isn't, you want to avoid it, but again, where we have really cold weather, do you know if it's okay if it freezes, just like the topsoil would on the ground, you have freeze events down to the frost depth, and then it comes back in the spring.
Mhm.
So I would kind of think that there could be, it's a little bit flexible. It just might take longer, but maybe you have different experience.
I would think that it would take longer, but in the ground, those worms are able to go down and stay out of that freezing. And that's what we're really concerned about, because the worms seem to be critical to the quality of the end product and the maturity of that product.
Another factor I want to point out is that microbes have this capability that they will form this hydrophobic layer around the organic matter. So if you let it dry out, or if it's kind of freeze dry, I'm wondering if that is going to bring it back, or you might have to take it apart and then shred the content again and re-soak and then basically rebuild. Because we heard from several people that after the freezing process that their product was not very good. They don't have the efficacy. And that would be really devastating if you just wait and wait and wait another year and ended up not having the efficacy.
If you want it next year at this time for planting.
So it sounds essentially to treat it like because we vermicompost as well. So it sounds like you should just treat it like a vermicompost and make all those. Yeah. Okay.
It is a vermicompost.
So where are you, Chris?
It's just north of Santa Fe.
Okay.
Okay.
All right.
Josh Waybright, do you want to ask your question about mob grazing?
Yes, I was wondering if I've got horses and 7 acres in Santa Fe, and I've been thinking about trying. I've been down and you guys, does it work the same way with horses? They're not ruminants, so I don't know if that matters or not.
If you handle the impacts the way the bison would have, it should. It's that impact that they have on the soil. It's the biology that comes in their dung and their saliva and their urine. It's the how much you let them graze. You don't let them graze over 40%. And that allows the system to feed the microbes in the soil and help them build up. But you should watch on carboncowboys.org and see the impact of these animals.
Yeah, you would start to develop this idea that all the answers comes back to it depends. So it depends on the type of animal, the way, the hoof impact, how they eat, what type of vegetation they eat, and then therefore how actively you have to manage them, and to make sure they move on to a different paddock or make sure that they eat everything. So there are a lot of moving parts there. So you have to be at the very beginning, at least, that you need to be very observant, so that what is the next step for you.
And each animal has its own impact on it. It's a system that we restore. It's not just the bison. It's the predators that keep them moving. It's the grouse out there that spread the manure. It's the dung beetles. It's the insect community. It's a whole system that we're trying to rebuild.
Mm-hmm.
And Josh, I will note that Alan Savory, when he lived in Albuquerque, had 2 or 3 horses on about a half an acre, and was able to achieve grass that was over the backs of the horses. So it's possible. Just have to keep at it and keep learning.
Yep.
Excellent. Thank you.
Keep observing.
Let's see. Let's go to Greg Cording.
Well, this is embarrassing. I ask questions I don't really understand, but
That's all right, Greg. I give answers I don't understand too.
Okay. Decades ago, I just had Bio 101 class, and she said, pick an organism and do a report on it. So I found Archaea, and there's European studies that show 2 times to 3,000 times more nitrogen fixing Archaea in soils of all types, wild soils, wildlands, farmlands, than nitrogen fixing bacteria. I'm just wondering if you look at those separately, or do people lump them together as just all the nitrogen fixing microbes?
Well, the analysis I do is metagenomic. And so we canvass every microbe in there, either fungi or bacteria. We usually see about 3,000 species of microbes, bacteria and fungi. So we're tracking everything. We're tracking archaea, we're tracking bacteria, we're tracking viruses, we're tracking eukaryotes, so all your fungi, all your insect communities. They show up in this analysis. So it's a complete system approach in that soil as far as the microbes.
Oh, okay.
And interestingly, we've compared our compost to compost made in Australia. And they're almost identical in both the metagenomic structure and population, but also in the metatranscriptomic or the metabolic functionality. I didn't have time to show those slides, but it seems like there is a common endpoint in biodegradation that to see, well, there was 4 compost in Australia and we had 2 here that were almost identical. And I've never seen that.
And keep in mind that they're at the sea level at subtropical climate. And we're here in New Mexico at around 4,000 feet above sea level, desert, high desert area. We use mostly leaves for those composts that were in the analysis. And over there in Australia, they use, I guess, the eucalyptus leaves and twigs and some maybe grass clipping. So they're different input material, different climate, and different elevations and latitude. But yet, the analysis came back so similar. And that gave us such a hopefulness that you don't have to take the original compost material from us that to inoculate anybody. That if you follow the method properly, you can get to the same endpoint.
Yep.
Good. And I think, Greg, the next comment here is actually from you about Ibrahim. Would you like to bring that up?
Oh, I'm sorry. That was for Chris. Ibrahim Lokes
Oh, okay.
has a place in Santa Fe, and he has several bioreactors outdoors, but he has one indoors in a hoop house. So he might have data on comparing the two for freezing. And
Yeah.
you can find him on Facebook under Mother Nature Center.
Okay.
And I believe he's on your register, David.
What's his
he's-
name
he's-
again?
Ibrahim.
Ibrahim?
Ibrahim Lokes, Loeks.
Right, right here.
Oh, okay.
Yep. And he's also done a half-size bioreactor, so which I don't think is produced yet. So.
Oh, yes, it has. It's amazing. It's
Has it?
just it's full of worms. It's alive.
Oh, great.
Oh, right.
That's what it's supposed to look like. It's supposed to end up looking like a deep, dark, rich chocolate cake. Full of worms.
Okay. All right. Linda Poole, you are, looks like you have the final question here.
Well, if I'm the final one, I just want to say thank you so much for this whole series as a whole, but especially for tonight and Dr. Johnson. And I'm sorry, sorry.
It's
Yeah.
Hui-Chun.
Hui-Chun. Okay. Well, thank you for this. I do wonder about the use of the bioreactor extract for rebuilding native vegetation. I wonder if when you're building the bioreactor, if I was to take clippings from the desirable species that I want to have in there and incorporate that with the nitrogen and the straw and, I raise sheep and that part, would it give it any more advantage for the use of restoring native ecosystems based on what I build it from?
That's a good question. And I haven't done the research, so I can't answer that one.
Yeah, we don't really know,
But we do have a job for you.
Well, I have one. I have one started, so
Okay.
I guess I'll know in a year or so.
Okay, but you need a control one also.
Yeah.
Ah.
Yeah.
Okay, and now I have to build another one. That's a good project. Thank you.
Sure, any day.
Okay, very good. Well, and so Linda had our last question. Isabel, did you have anything, the date maybe for our next soil stories that you would like to announce?
Yeah, absolutely. This was fascinating. Thank you both so much, David.
Thank you all
Hui-Chun.
too. We so appreciate you getting this out.
That was really, really wonderful. And to the whole community tonight.
Yes, thanks to everybody for coming. We really appreciate the interest, and let's go make this planet better.
Well, thank you all very much.